This is a presentation about point mutations by Mackowsky. Point mutations result in the substitution of one base for another. There are two types, transition and transversion. Transition involves the substitution of a purine for a purine or a pyrimidine for a pyrimidine. Our purines are adenine and guanine, and our pyrimidines are cytosine and thymine, shown in the examples below on the left. On the other hand, a transversion involves the substitution of a purine for a pyrimidine or a pyrimidine for a purine. We are given the coding strand of DNA, and from this we can determine the DNA template strand, the mRNA strand, and the resulting amino acid sequence. We will use this as the basis for our normal DNA sequence. We will now look at what various point mutations would do to this strand of DNA and its resulting amino acid sequence. The first type of point mutation is a missense, in which the point mutation changes the resulting amino acid. This can either affect the function of the protein or be neutral, in which it does not affect the protein's function. Let's substitute the second guanine of the coding strand with a cytosine, a transversion. This point mutation then changes the resulting mRNA strand by changing UGG to UCG, which then changes the amino acid sequence because UCG codes for an entirely different codon from UGG. Tryptophan is changed to serine. The next type of point mutation is a nonsense mutation, in which a codon is changed to become a stop codon. Let's substitute the second guanine of the original coding strand with an adenine, a transition. This point mutation changes the resulting mRNA strand by changing UGG to UAG, which then changes the resulting amino acid sequence because UAG is a stop codon. Next is the silent point mutation, in which the substituted base still codes for the same amino acid. Let's substitute the third thymine from the original coding strand with a guanine base, a transversion. This point mutation changes the resulting mRNA strand by changing GCU to GCG. But it does not change the amino acid sequence because GCU and GCG both code for alanine. Lastly is the read-through point mutation, in which the stop codon is changed to a codon that codes for an amino acid, resulting in a longer protein. Let's substitute the fifth thymine with a guanine, a transversion. This point mutation changes the resulting mRNA strand by changing UGA to GGA, which then changes the amino acid sequence. The stop codon from the original sequence becomes glycine, and the rest of the mRNA strand can now be coded for more amino acids. 2 more amino acids are added to the amino acid sequence and the protein becomes longer. Here are the references used in making this video. Thank you for watching.